Thank you for having this golden opportunity to contribute to this exciting area and exciting industry today. Since I'm neither crop scientist nor computer scientist, I will start with a brief introduction of my background, how I appear to be in this space. And then I will introduce the Ag ImageNet that we're developing here with the Image Formation and Processing Group at Beckman Institute, that I believe will be the key to transforming the global agriculture. I will talk about my collaboration with the Professor Nico Martin from Crop Sciences, and I will briefly summarize with the developments at my company to the extent that I can share here publicly. So I got my PhD in math in Russian Academy of Sciences in Moscow in 1992, upon which I assumed a few postdocs positions in Germany and France. Then in 1998, I landed at Georgia Tech as a visiting scientist in a flight control group, got familiar very well with the aerospace industry, their challenges. In 2003, I assumed the faculty position in Virginia Tech in aerospace and ocean engineering. In 2008, I moved to Illinois, and in 2014, I co-founded the company Intellinair. So what you see here in these red frames are applications and transitions of technology that I have developed in my academic lab, so to say. In particular, this is all focused on the control technology that we were able to flight test on subscale commercial jet, to fly on Learjet, F-16, to commercialize it for Raymarine Evolution Autopilot, for hydraulic pumps in Caterpillar, and we went through experimental rig tests at Statoil, Norway, although with the oil prices crashing, we couldn't get into production. So this is maybe a picture of 2013-14 when the oil markets went down. So having so much success with different industries, I thought also about starting a company on drone technology, since drones were kind of going to hit the consumer markets. And being in Midwest reality, so the first investments came from agriculture. So the agricultural vertical quickly took us, and and moreover, not just investments came from farmers, but also we got lots of farmers who offered their big farms for data collection. And very quickly it became clear that the sensor package that's needed really for delivering actionable insights to the farmer, and not just the mosaic maps of their farm, can range up to $250K, okay? So such a sensor package cannot be put on a drone and flown. So we started flying piloted aircraft. We are already 5 years in business, 3 years with farming customers. And as I said, the main challenge that we're trying to solve for farmers, to bring them actionable insights. So we have about 300 terabytes of high-resolution data collected from Midwest soybean and corn farms, and just following the developments that the ImageNet generated through different industries, we started building the agricultural ImageNet. I will talk about it more maybe towards the end of this presentation, but the 300 terabyte of data gives us the hope that we can come up with this structured data set with labels and annotations that can have the power to transform the global agriculture. So, as I said, I want to talk a little bit about my collaboration with Professor Nico Martin from Crop Sciences, where we are working on optimization of nutrient management by building deep neural networks and predicting the yield after we have the trained models. So we use the data from data-intensive farm management project of University of Illinois that Professor David Bullock has kind of led that project. So we have access to data of 9 research fields. And we just summarized our first paper, how we modify, for example, the existing deep learning algorithm with added momentum term to achieve the best kind of optimization, to have optimal fertilizer maps. And as I was told that this is going to be a 5-minute talk, I'm at 4:30 mark. And this is the last slide of the Intellinair app. So basically the company, as I said, it's already the 3rd year with customers. We have really big customers. The app basically does not just show them the insights, but it also organizes the alerts to simplify the farmer's job. Because we do have farmers who have big farms that we are servicing, like more than 30,000 acres, and this year actually we have 1.3 million acres in service. So we organize also their alerts by the order of relevance. They may have a small area with weeds, for example, that may matter a lot more than another area, bigger area with width. So understanding the farm history, the data availability, the farmers' insights priorities. So we have a rule-based system according to which we organize the alerts to serve so many farmers. And here the piloted flights are crucial to scaling the business model, because if we go with drones, we can collect all the data, but maybe not at the same time of the day, not with the same lightening and shade characteristics and so on. Analyzing this type of data, especially when digital agriculture, I think, is a relatively new industry. There isn't a big history of having this type of data, organizing this type of data, managing this type of data. So to make the model scalable, indeed the business model lies at the core of how we drive the scientific discovery forward. So this is just a big overview of different apps. This is the light scheme. We have also the dark scheme version of this app. And with that, I'm at 6-minute mark, so I'm happy maybe to take one or two few quick questions, right? 5-minute talk lasted 6.
I have a question.
Uh-huh.
What is the scale of the data, the amount of data that you're working with, and do you expect the volume to be much higher in the future?
Sure.
And what kinds of things what kind of challenges then would come up if data became?
Well, our customer database has doubled and tripled over the years, and that implies that we're getting more and more data. Like last year, this time we were talking about having 150 terabytes of high-resolution data. Today we are already analyzing 300 terabytes of data, and by the end of this season it will be a lot more. So indeed, the challenge with data collection, with the current business model that we have, we deliver piloted flights. 13 flights through season is what we commit to the customer, and within 24, 48 hours he gets the alerts here, organized set of alerts on his app. What will happen in the future? So, in the future is this: the satellite data gets cheaper and cheaper. Can we get the resolution that we get today from airplanes? We get 10 centimeter per pixel. Drones were doing 5 centimeter per pixel that we used in the first year to get started on the algorithms. But the way the farmers grew in this business, the number of farmers, we need maybe millions of drones to cover all of that at the same time of the day with all these sensors, and so we switched to pilot it to make the business model valuable, right, viable, so that we can collect it at a cost that's affordable for the farmer. So today we have roughly $34 return on investment for the farmer already estimated. Over the years, my feeling is that satellites may eventually get higher and higher resolution, but will they provide thermal imagery? Will they provide all these kinds of other sensors that we have? No. But we'll get more data from soil maybe. More data will come from ground robots, combines, and so on. And layering all these different layers of data should enable maybe to reduce the role of aerial imagery to some extent, or maybe organize how we collect the data. Now we do 13 flights. It's a subscription service. But over the years, by learning the history of every farm in detail, some farms may need a few flights. Some farms may need a lot more flights, right? It's like when you go to a doctor, right? You do your vitals. That's the scalable model, right? They get blood count and everything. But then if you have some issues, they may do you more detailed tests more often. So it's like preventive medicine, basically. Alert system. Thank you. So your slides are here?
Thank you, Naira. So another change to the schedule, we're going to have Caillou Guan speak next because he needs to leave soon. So Caillou, thank you.
I'm going to shift the gear a little bit. I talk about satellite and talk about my work that I've been working on with Professor Champaign from computer science in the past 3 years. So I actually prepare a little bit more slides, and so I probably can fly a little bit faster. So I think the program that we really think about is supercomputing satellite. Supercomputing satellite is primarily our tool, and agriculture is our domain. So my work with Jen mostly focus in the Corn Belt, and the importance is needless to say here. So I think the biggest problem is some of you already very much know is satellite data is either have a very fine spatial resolution but very infrequent, or you have very frequent image but very coarse resolution. And usually this is a standard trade-off. Now you have some commercial satellite data like Planet Lab, but what we are trying to come up with is we really want to build a very advanced fusion algorithm, and so we can fuse all sorts of the satellite data together to generate very high resolution map at the daily scale. So what you are seeing here is the Champaign County that from 2017, from May to October, it's actually fused at 30-meter resolution. At daily step, you will see all these fields pop out, reach to the peak time for both corn and soybean and start to senescence at different pace and harvest at one point. And so this can be done only with the power of high performance computing in blue waters as well as probably in the commercial cloud system and to scale up. But we have done this for multiple field major states in the Corn Belt starting from 2000 till today. And so basically the strengths that we have is for every individual field you want to look at, for example, these two individual fields, you actually can go back to the time and look at the individual progress over the whole time at the resolution of 30-meter resolution. Now, it definitely allows you to do something, but you can do something, you could not do something, right? So what we can do is we can easily identify the crop types, and that's actually have been pretty much achieved. We can now do very large scale in season in crop type. But also another thing that we have been heavily invested on working on is really come up with algorithm to really predict crop yield. So for example, we actually can do every individual field's crop yield in Illinois. You actually aggregate to the county level, you find it's very consistent with the USDA prediction. It's all out of some prediction. And then 2012 is a big drought, and we find we can also do that. And the similar pattern, we can do that actually for the whole Corn Belt. And so this is a paper that recently published. We actually can use a similar approach that do at the national level for corn. And so this is a new thing that happened in my lab. So I think over the past 3 years, in my lab and Japan, with some collaborators inside and outside of campus, what we really did is we use satellite data. We actually have a pretty rigorous idea, robust idea about what's grow on the ground, what's the biomass on the ground, even what's the yield. And we also have a new product about evapotranspiration at 30-meter resolution daily that we can do that for the large scale of the Corn Belt. Now what's next is we have a lot of stuff on above ground, but what's mostly intriguing, as everybody knows, is below ground. And so all inside the canopy. So what we are trying to build up is really tackle some of the most challenging questions about nitrogen, about soil moisture inside the soil. And so if we talk about soil moisture, the most outstanding feature that we have is tile drainage. Some features like this, and how are we going to deal with that? And this is some of the big puzzles that we have. So our solution is sort of integrated. We know this, but we don't know the hydrological conditions, we don't know the nitrogen conditions. So what we come up with, we use a lot of the satellite data that we already have to constrain some of the best process-based models, but then also start to work with sensor people, IoT people, to have some sensors at the locations that can further verify some of our understandings below ground. So some of this work has been recently funded by NSF Career Award. So and so I think I think the collective strengths that we really have is we have the domain, we have the CS, we also have the NCSA. Very proud to have these resources that allow these to happen. And I think all these expertise actually are needed to integrate such that we can do meaningful things. And so this is some of the state mission statements that I motivate my group, that we can really hope to do every field at real-time monitor and model the whole growth for the whole system. So I'm talking about the whole system, not individual field, not surface. I'm talking about the holistic system. But we hope to, down the road, we can really achieve the co-sustainability of both environment and food security. So with that, thank you.
So this GEOSYS is doing this work of merging different data from different satellite sources. So what would be, they're looking at, I think they say 13 different sources. So how
No, it's 30-meter resolution daily. Sorry.
Also, what Geosys is doing. So Geosys, there's this company that was owned by Land O'Lakes that just got sold to Earthcast. So their main value add is they look at data across, I think it's different, I don't know how many, I think between 10 and 13 different satellite sources, and they merge it. And they provide, I think they said 13 clean images for an entire season. So I don't know if you've looked at them, but that would be something to look at from a commercial side.
Thank you. Yep.
Thanks, Caillou. And so our amended schedule, I think we have Andrew Leakey next. Andrew, if you're able to come up.
I want to try to convey a little bit of what I think some of the challenges and opportunities are for us to work within this center and bring a plant science perspective to that. And in particular, one that relates to crop improvement, not just production agriculture. I think we've had a bit more focus on, what kind of information can we get about farmers' fields. A lot of what I focus on is what can I learn about how plants actually work. Those are very related problems, but they're also somewhat distinct. A key element of this is on the biological side. We're really interested in understanding mechanisms across scales of organization, and that's what I'm trying to convey on this slide. So at the bottom left-hand side, down in the corner, that's the signal transduction network of a series of genes which are expressed to control the way that pores on the leaf surface develop. So you start off with an undifferentiated mass of cells, and you end up eventually with a leaf with all the cells in the right places, and there need to be pores on that leaf surface in order for carbon dioxide to get in and be captured by photosynthesis. So we're looking at molecular and genetic data. We're trying to link that, and this is one of our phenotyping problems, to microanatomy. So the next image you see is a scan of a leaf surface with the pores on it. Then I've zoomed in to show you what a single pore looks like, because we take measurements of the gas fluxes actually going in and out. And then lastly, we want to be able to mathematically model that. So I've given you a nice biologist's simple straight line equation there. And then ultimately, we want to actually understand how this relates to crop performance in the field. And so this is a thermal image from one of our genotype experiments, looking at variation in canopy temperatures, which tell us something about how much water the crop is using. I threw this in because I wasn't sure how much background we would have had on the sort of whole system, but I think Vikram and Matt have given you a real sense of the diversity of computer science and engineering tools which we need to address this goal. But so I won't repeat all of this, but I just want to tell you that this was the vision that Joe Cornelius put out there when he was leading ARPA-E to really invest in this area. And I want to encourage those of you who are at the north end of campus that not just ARPA-E, but a lot of other agencies are really enthusiastic to invest in this. And those of us at the south end of campus have been engaging NSF, the Department of Energy, USDA, Bill and Melinda Gates Foundation to work on this. And we're collecting a lot of data, but often we're limited by the tools that you can provide us. And there's a big community of us that will be excited to interact with you. To get back to my sort of initial theme, this is a range of what we call phenotyping techniques that we're trying to put together across lots of scales that I described a little bit before. But part of what I want to emphasize is one of our big problems, at least within my research group, is computer vision. So you can see we're gathering images in the bottom left-hand corner at a micrometer scale of what the leaf surface looks like, and we want to be able to extract information and objects out of those. We've got very literally and figuratively dirty images in the orange there in the middle at the top of where the roots are actually in the soil, in the field, hundreds of thousands of those, which, and you'll be horrified by this, traditionally undergraduates have manually drawn around these. We're talking experiments that take person months of very laborious analysis. We have hyperspectral data that is informing us about the structure and the chemical composition of leaves, which we're really interested in. And then we're taking images both from drones and also from rovers below the canopy that tell us something about how much water the crop is using, how much biomass is being produced. In a number of cases, we have early phase products which are helping us analyze this, but there's a almost limitless set of questions that we have, and we'd be really enthusiastic to interact with people on this. I want to just give one example. So we're really interested in these pores that are on the leaf surface that impact how much water is being released. And so we've developed a pipeline where we're using optical tomography to scan the leaf surface with a laser. You can get a three-dimensional reconstruction of it like you see in that image. And we've been working with collaborators at Wisconsin to build a pattern recognition neural network to count those pores. It's by no means perfect at this stage, but we're getting reasonable correspondence between human and computer counts, and that's now allowing us to do genotype to phenotype associations and try to find the genes that control this response. But counting is simple, relatively simple, and not all that informative. As a physiologist, I want to understand how big those pores are, what the pattern is with which they're arranged across the leaf surface, and all sorts of other pieces of information that we definitely don't have the computer vision tools to pick out at the minute. The other thing I want to emphasize, if you're in that area, that makes biology interesting is counting those objects on these images looks relatively straightforward. This is the slide I show the biologists when I want to convince them this is possible. But this is the slide I'll show you to describe that in a biological context, we're searching for an object within these images, but there's incredible diversity across even just corn in what the leaf surface looks like. So this is 4 different varieties of corn. All of them have those pores on them. In an example like this, you can see them really easily, and I've just highlighted them with those triangles. But if I go up to TZI 8 at the top, they're almost completely lost in there. They look the same if you know what you're looking for, but the context in which we're hunting for them can be really very variable. And I'm hoping that's the kind of challenge that makes this appealing to those of you out there who think about this kind of thing. Just to change scale completely, we do thermal imagery from the air. And so this is an example of a field with nearly 1,000 plots of sorghum in it, and we can pick out lines that have high or low temperatures. But thermal cameras are really limiting in terms of being low resolution, requiring a lot of extra hardware wrapped around them to maintain stable temperatures and so forth. And we'd love to talk to engineers about ways in which those could be potentially improved, just beyond the image processing problem. And I want to convey that we can take this information and then try to make plants that actually perform better. And so my RPE project is called West, and this is just one of our preliminary results where we've transgenically made sorghum that has fewer of those pores. And the result, if we then expose it to drought stress by not watering it, you can see that the water-efficient sorghum on the right-hand side is growing nicely and is large and happy, whereas the standard one on the left-hand side is wilting and drought-stressed. And so we're trying to wrap these tools together to lead to these sorts of improvements. I'm running out of time, and I just want to finish with this. The most significant activity I've been involved in with respect to the center so far is there was a large group of us, about 26, who last month submitted a pre-proposal to NSF. They called it a pre-proposal, but it was probably about 50 pages long in the end. And it was for this new NSF mid-scale infrastructure program that they're running. And so we've asked for $20 million to try to bring together a set of things that Illinois is uniquely experienced and capable of, but which have never actually been combined before. So those of us on the south end of campus run a facility called Soyface. This field that you can see in the picture is an aerial shot of that. It's essentially an outdoor laboratory for studying how crops will be impacted by climate change. It's 80 acres. You can step into plots where plants are growing at the CO2 concentrations or the air pollution conditions of the future. We can use retractable roofs to catch all of the rain while still allowing the plants to grow in the field, experiencing drought stress. And what we want to do is overlay that with phenotyping from autonomous drones, rovers, as well as a large cable-mounted camera system. And then we want to wrap that all up with the cyber infrastructure that NCSA can deliver and the sorts of exciting computer science tools you've heard about from other members of the center this week. And so this is a challenge that spans bioinformatics, biology, computer science, cyber infrastructure, engineering and robotics, sensors and imaging. But I'm really excited that, here as an institution, we're uniquely placed to put all those things together in a way that few, if any, other places are able to do. Cool. Thank you.
Questions? Do you see a role for something like farm beats in that mid-scale infrastructure?
Oh yeah, something like when I heard that was amazing. If we could just pop that on top of the field site, and then suddenly that whole heterogeneous mix of sensors that we have out there could be connected to it, that would break a huge bottleneck. Yes.
Thank you, Andrew. Okay, next up we have Ludmila Meinzer, Technical Program Manager from NCSA. You may remember from yesterday.
Thank you, Matt. I think since yesterday I've come to be associated with chocolate cows. I do like chocolate, and I like cows, both live and in the beef form. Okay, so now that you got your pre-lunch dose of levity, we can go on with the featured presentations of today. I will be discussing the uses of high-performance computing for agriculture, specifically in genomics, although we do genomics in areas outside agriculture as well. So throughout yesterday and also this morning, many times people have mentioned how we need the cyber infrastructure to do the highly computationally intensive things that are required to move digital agriculture forward. And hopefully the talk will cover various aspects of what gets involved. For background, NCSA Genomics, which I lead, is a team within NCSA Industry, which combines multiple domains of expertise: multiphysics, computational fluid dynamics, data analytics. We work with industry partners, which is to say companies, some of them listed here, but we also work extensively with UIUC faculty and also professors outside UIUC, just because the field is developing so fast and is so fast-paced, the work, that we simply cannot maintain the necessary expertise unless we're engaged in research projects on the cutting edge with faculty. So the work is highly multidisciplinary, which requires a multidisciplinary team of people from all the different backgrounds, from math and stats to computing, crop sciences, bioinformatics, information management, physics. And together we're engaged in a very diverse range of projects, sort of cross-pollinating each other and also with the faculty and the industry partners. And these projects range from IT, which we will discuss a little bit today, and that has to do with hardware, software, data transport, data management, also software development and optimization projects, as well as the scientific research projects, as I mentioned before. So let me just showcase some examples of these to get a taste of what we tend to do. For IT projects, what we tend to do a lot, and this is, I know, very boring and mundane, is software installs and configuration. The reason why this is important is because the field of bioinformatics, as I say, is developing very fast. And we're still working out, what is the appropriate algorithm to do this thing. And nearly, every month and sometimes every week, someone comes out with a better solution to the same problem. So you not infrequently will have dozens and sometimes hundreds of software that can do the same thing. So someone needs to know how to handle it, how to use the software, how to evaluate whether it is in fact better than its competitors. And as a result, we take care of installing all these different software packages on the high-performance computing system maintained at NCSA for industry. At the moment, we have over 200 software packages. We've accumulated them only over the course of the last 2.5 years. If you look at the omics tools, which is the group that sort of scours the net and keeps as complete list as possible of all the software available, out there in genomics alone, there are over 26,000 different software tools. And of course, everybody has their own favorite thing. Anyway, here we are. If you want software, come to us. The thing also about software in genomics and adjacent disciplines is invariably every analysis is a workflow. of workflows. And these are very complex nested beasts which are difficult to manage. Number one, to install them, every single module in the analysis pipeline is usually software with multiple dependencies. Number two, when you use a workflow and your data have to come through one step at a time with one tool producing outputs that serve as inputs to the next tool and so forth, it becomes difficult to manage on the cluster when you have to run it on very large sample sizes. So for example, if you have to do an analysis on one sample and you put it through a workflow, well, that's maybe one job on one node. And if you have to do analysis on 100 samples, well, that is 100 jobs across 100 nodes. And things become more complicated if your workflows have fans and merges, so that some steps maybe have to be parallelized across multiple nodes. Then it becomes messy very quickly. Not to mention all the intermediary files that get created at every individual step in the process, and that could give a hit to the file system. So here's an example that should be near and dear to some of you in genomics. On the left is Maker, our favorite. We set it up in 2016. It worked just fine, and then we upgraded the system software stack on the cluster, and it kind of broke. We thought we kind of fixed it. We really didn't. Now we have to rebuild it. It's a pain. But because we stay in practice and do this every day, we could be able to help you out if you have a similar problem for your area. Number three, we do a lot of hardware benchmarking. This here has to do with hardware purchasing decisions. It is an example for industry. If there was a company that maybe was trying to decide what is the new cluster they need to invest money in, and we could go and figure out their patterns of job submissions and how their software runs, and then look at the newest hardware that is about to come out to the market, do our benchmarking, and make recommendations. And I know that this audience is mostly academics, but I think it is still relevant because just yesterday we were discussing here in the panel about bringing compute to the data. Well, in order to figure out what kind of compute we need for a particular computational problem, we need to do hardware benchmarking. So there you go. In terms of the software development projects, we do a lot of performance benchmarking and improvement. Anything from just figuring out whether the software is optimal and could scale to actually improving software scaling and parallelization. We also develop new software. Here's an example from genomics that is applicable to both ag and medicine. 2 minutes, no problem. It has to do with the genome-wide association studies, specifically including epistasis. In genome-wide association studies, we're trying to find the connection between genomic variants, mutations, and phenotypic features. But the problem is that the genomic variants frequently work together because they're not just some letters that are floating around in your cells. They're part of genes which get translated into proteins, and proteins interact with one another. So you have to consider actually networks of things, and this is a complex computational problem. It has not been yet solved for interesting phenotypes, which is to say continuous phenotypes that are determined by multiple mutations. We're writing software to do it from scratch. Okay, and finally, the science project examples. Here is one, again, that is applicable to both ag and medicine and has to do with structural variant calling, which is more difficult to do than detection of individual single nucleotide variations or small insertions and deletions. This has to do with actual large insertions, deletions, and inversions. And in order to do that effectively, one needs to basically construct de novo genome assemblies and then compare them among samples. This is particularly hard in plants because there's polyploidy that prevents you from constructing a good genome assembly. There's also duplication events within the genome. So we have been working for several years now to construct a workflow around a piece of software that is capable of doing so. And one final example that also speaks to the panel yesterday, data harmonization. When you have an area that has to combine information from genomics, metabolomics, socioeconomics, phenotypic factors, GIS, how does one combine these data together? How does one do cleaning in an effective, automated way in order to result in quality analysis? It's complex. Here's the answer. The end.
When you say if we have software needs come to you, do you mean come to you to collaborate or come to you to pay you?
Are you a faculty or a piece of industry?
I am a faculty.
Okay, then we collaborate.
Great.
By writing grant proposals together so they can pay the both of us. Any other money questions? No? Okay.
Thank you, Luda. Gary Schnitke is up next. I think, oh, there. So, and then we have Rob Cooper and David Bullock.
Good morning, and I'm going to just a little background here. I'm in the Department of Agriculture and Consumer Economics. I'm actually a farm management specialist in that department, and I have an extension appointment. And I do a lot of work with farms in the state, do a lot of research on farms, and get a lot of questions. One of the things that has bearing on the digital center is our relationship with Illinois Farm Business Farm Management, which informs a lot of the decisions that we provide to farmers. But that is a financial record-keeping and advisement service that runs in the state. It has about 6,000 farms enrolled in it, and it represents about 25% of the acres in Illinois. It's unique. It is the largest farm-level data set in the nation, and it's is the best, I would argue. We actually have a SQL database with 1,000 elements per farm. It all relates to revenue, expenses, assets, liabilities on those farms. So we have a good picture of what those farms are doing. We don't have the transactional database readily available from that. We have the summarized data. Transactional database exists on individual farmers' PCs. They're generally software, accounting software that we then summarize, or FBFM summarized and provides that summarized database. One other thing is that many farms also in Illinois have spatial yield data, spatial soil and application maps, and maybe on a number of large, or whatever you want to call them, big data websites, Climate Corp, Captivia, you name it, they're on it. We actually do not make any of use currently of that data when we're doing financial analysis. Type of questions I get are expansion and scope decisions. How should I be expanding this farm if I want to? And how fast? How do I wisely acquire capital, debt financing, risk management, crop insurance, and those type decisions? Sort of a corn, soybeans with crop decisions. The fifth one there, machinery and tillage decisions, those are fairly complicated from a farmer perspective. They involve, obviously, capital, large amounts of capital, and often involve changes in the operational structure of the farm, timing of farming applications, and then farm transition decisions. All of those, you'll notice two things about those. They involve risk. So you make those decisions, the outcome on the farm isn't known, and there's usually a time element to it too. You make a decision, it's going to impact decisions in the future. In my opinion, one of the, those production decisions that only have one within-year impacts are easy to evaluate. These are much harder. All right, so what we would like to do, here's where we want to go. Here's questions that, and I got the three. All right, and this is the question the farmer would ask, did I have a good year or was I just lucky? All right, let me give you an example. As part of FBFM, they get benchmarking reports that say you were X% better than average or X% lower than average. there's always a certain amount of randomness associated with that. And just think yield variability within county, within the state, there's a tremendous amount of yield variability, and marketing also has quite a bit to do with this. We could answer that question if we could link up those farms' financial data and have an understanding of where their farms are at, the weather patterns that exist on those farms, the soil productivity rating on those farms. And the more we know about the production practices, the more you could answer that first question. Did I have a good year or was I lucky? Second thing that we would like to do is have closer to real-time risk management. Tomorrow, Scott and many other people are going to have to make a crop insurance decision. It would be nice to be able to say, right now we provide county level case farms. It would be nice to have that to the farm level. This is what you farm, this is the risks that you face, here are the impacts of those decisions on that farm. And now let's take it in July. We've had all of this weather transpire. Here's now your yield distribution, and here's some of the things that you can think about. We would also think, and these are all questions that we get, economics of tillage and management systems. It's hard to imagine the number of different tillage systems that are out there. In fact, if there's 60,000 farms, I would guess there's 40,000 tillage systems out there. Everyone seems to have their own unique one. But it would be nice, and all of those have, obviously, some impacts on the economics. We would like to be able to use that data, and again, that revolves around a lot more linking of the production type data that exists that we aren't getting, and better evaluate those decisions. The fourth one in red is where I think we would really like to go. Rather than having productionists say, we can do this, then we sit back and evaluate the value of it. And you go, yeah, it makes money, no, it doesn't. We would like to be exactly opposite. The economics saying, this is the technologies that we would like to see developed, and have it work the other way. So that would be the long-run goal, and I think we have opportunities to make strides in those areas. So I'll stop there and take any questions.
I think that's a great way to think about it. And I think one of the things that we need to do in the workshop is to, not workshop, the center, is to sit down with people like you who understand the economic challenges, the economic goals, and then think backwards about what problems farmers face and how technology might be able to help solve those problems.
And it could very well influence the type of decisions that in the end are made and the types of technologies that are in fact looked at. Yes, that's.
Hey Gary, with those huge data sets, is there anybody doing any predictive analysis across or machine learning, or are they you guys just doing standard economic analysis again?
So we're doing standard, so if you're looking at financial data, there's sort of us doing sort of economic analysis. The other group that makes use of larger data sets of farms are actually lending institutions and credit institutions, and they're evaluating risk. They're saying they want to know what their portfolio is doing and looking at portfolio risk, and there's a lot of regulatory. So those are the two types right now that are specifically that I know of that are looking at financial data. And again, it's sort of this credit, and we do standard, standard sort of things. So it would, yeah.
It strikes me there's a bit of an asymmetric information problem, right? Because the farmer has less information than people like the insurance brokers, the commodity markets, Climate Corps. Is that something you think we can address?
So the informational asymmetries are sort of interesting, and who knows what. Actually, the crop insurers often argue the farmer knows more than they do, but and then they're making adverse selection decisions. One of the interesting things is that you'll notice that we have financial data, and farmers are very reluctant to give that to anybody. Because the minute they give it to somebody, they're afraid that they're going to. That's why you hear the data concerns. All right, Monsanto gets my data. All right, they know everything about me. Are they going to use that? They're obviously going to use that to make more money for themselves. Is that going to benefit me? I mean, so the asymmetry of information as well as the motives. I understand the farmer's motive that I'm a little bit afraid of giving Monsanto, Microsoft, all my data, and then they sell me a prescription back and I lose my market advantage. And you have to be cognizant of those concerns.
Thank you very much. Okay, Rob Cooper, lead research programmer at NCSA, is up next. And
okay, so my name is Rob Cooper, and it's been referenced already by a couple of people, and I'll be talking about the TerraRef project in Arizona specifically and how we help them. It's not just me. We are a larger group. We do research and development here in NCSA. We have about 20 full-time people. And I like to see us as doing applied science. We sort of live between the researcher and the company, and we take some of the ideas that they do, and the scientists in a different field, some of the computer science ideas, and moving them into their field. So we help them a little bit. And so what we try to do is basically build frameworks that we can easily apply. And so one of the places where we did this is in the case of the TerraRef project. We have a scannalyzer, that's the picture that you see on the left, in Arizona. It's a humongous machine. The field is about 200 meters by 20 meters wide. Just to give you an idea of perspective, that little green box that you see on the right side is a full-size shipping container. Right? So that is the size. The thing is 3 stories high and spans the whole field. The little white box that you see hanging there is the actual sensors. This is the box where they can place any sensors in. And so the nice thing is we have a set of sensors in there right now, but we can add new sensors to it depending on what we see and what's happening. Right now, the plot itself that we capture on is 20 meters by 200 meters. They're doing the final set on adding another 200 meters to it, so we're basically covering about 2 acres with this. The sensor itself scans the whole field every 24 hours. So every day we get data. So to give you an idea of some of the sensors that we have near-infrared, visible light, IR, LiDAR, RGB, stereo cameras. All these cameras are basically sitting in this box and capturing data. We basically capture about 5 terabytes to 10 terabytes of data per year right now. With the addition to the field, we're moving up to about 10 to 20 terabytes of data that we need to process. Right, so I love Ranveer talking about the white band, but we have 5 terabytes to 10 terabytes of data that needs to be transferred. So there's your challenge for number 1. These cameras take high resolution images. So here's an example of what we get. Right? This is sub-millimeter accuracy on plants that we are collecting. We're taking all this data, combining it together. LiDAR data, we can get the plant height, so you can see there. Here's the pipeline that we do. On the left top side, we have the field. We use Globus transfer to transfer the data over to NCSA. We do minimal transfer at the Arizona site. We have a 70 terabyte storage. This is just to basically capture the fact that the internet will go down at some point. Something will happen. Somewhere something will go down. Like one thing that happened during the building of the extension is they actually cut through one of the fiber lines that we have running to the site. So we lost a couple of days where they actually had to reconnect the fiber back up to get the data. So we have about 1 to 2 weeks of backup that we can store in Arizona before we actually start losing data. Because plants don't stop growing. right? Most people will know that, but this basically is something different. I cannot stop my simulation. I can stop a simulation for a little bit if something goes wrong, but I can't tell a plant to stop growing, please, because my disk is full, right? So we don't have that luxury. Once we're here, we land the data in Clouder system, and this is something that we've developed, we've used in many different projects, and this is our data management system. Once the data is landed here, we basically trigger additional processing from happening. I'll go into that in a second. Once all the data is processed, we basically upload the data back as features back into the Clouder system, as well as into the Betty database system, which is a phenotyping system that we have developed as well. So the Clouder system itself, it's highly configurable, and it's basically we use in a lot of places. And so the nice thing here is that we have the system where the data comes in, and every time data arrives here, we start an extraction event. We say, so a new file arrives, we start extracting information from it. This basically makes it easy for us to take new algorithms that people are developing and plugging them into this existing infrastructure. So if I have a new algorithm that comes in, or a scientist develops a new algorithm, I don't have to rebuild this whole new architecture. I just deploy a new Docker container, and it becomes part of the whole infrastructure. And so what we have is we have these complex pipelines. So the orange are the data products that we start with. That transfers over. We basically convert the RGB binary data into a geotiff image which the scientist then can start using. We take all these geotiff images and stitch them together in a full field image. Right? So we're going from a regular small image, which is just your standard camera, a couple of megabytes, to a full field stitched image, which is about 250 gigabyte size. Right? So now we start actually talking about large amounts of data. In the end, once we have the full field, we can extract the canopy height from these full field images, and we take these traits and store them as phenotypes in our database. And so this allows us to eventually have these phenotypes that we extract from this raw data associated with the genotypes that we know from the plants that were planted in the field, and basically start to do some analysis. How does these genotypes affect the phenotypes that we collect? And so that's where we really want to. So I'll give you an example of a full field image. Here you can see the whole full field. You can see all the different plots that are sitting there. You can see some plots are doing better than other plots. So this is basically, we have different genotypes, different treatments that are applied to all the different plots. There's about 2,500 separate plots that we have on this particular field. Let's see. Just kick me off when I run out of time, because I have more slides. This is our data. You can see we have about 800 terabytes of data right now. So this is about 2.5 years of data. We have about a petabyte of data that we basically make available for people. So this is to us the interesting thing is, right? This is where I say we work together with computer scientists. We make this data available. Like the next day, you can see the data from yesterday. That's already in the system at that point, some processing done on it. And so you as a computer scientist can then come in and basically say, hey, I'm interested in this data. I'm building a new algorithm. We take that algorithm, move it part of our pipeline, started extracting the phenotypes from it. And then as the agriculture person, you can come in and say, hey, how does this wonderful new algorithm that was developed, how can I look at these phenotypes and see how they match up with the plants that I know? So I can go to my specific experimental plot and say, what new data is available today? How are my plants growing? And so we can use that information to basically come up with better models for the plant growth, as well as for the scientists, the agriculture scientists, say, hey, this genotype is doing really well. I want to continue growing with this particular one, or continue breeding with this one. This one, like if I go back to my full field image, for instance, right? I see some of the bare ones. Those I might want to throw out, because those genotypes apparently are not growing very well. With all that data, we have 36 million files. Accessing the data, organizing the data is important. This is where we use the Clouder system for. We organize it by plot, by space. Data can be in multiple different locations, so it allows the user to easily find the data that they're interested in. We use standards for this, so that basically you can use machine-readable processes to get access to the data. We store as much metadata, so we store when it was taken, where it was taken. We store the exact location of the gantry where the picture was taken, so you have all that metadata as a scientist immediately available. So you can access all that data, look at it, and use that to develop your algorithms to hopefully help with better plant access. And this is the final slide I want to show quickly. This is the phenotype trait system that we have. We store the exact plants, the treatments that they applied to those plants. Did they give them water? What other treatments do we have? Do we actually have, some of them we actually have field hand-measured data. So the scientist actually went into the field and measured all the plants and the leaves, etc. And we have all that sitting here. And that is basically immediately available. So anybody can come here, and this is just open data in that sense, right? You can access this data and look at it and use that in your own research. So we have about a petabyte of data. This is something that we would love for people to start using, to start upcoming with better algorithms to help with the prediction of the yield of these plants. So, questions?
If you have hundreds of little plots there, do you have somebody bringing in a certain kind of soil with certain properties on 100 different places? Because if it's all the same soil, what's the inference space?
So the difference is the different genotypes that we plant. So we plant different plants there. They're all sorghum, but they're all different varieties of sorghum that are being planted, as well as the water treatment. So we have specific water treatments for each of these plots, so we can add more or less water to it. I do not know whether or not they use different soil or whether they use different.
Okay, thanks.
But yeah, but all that is captured, that could potentially be captured in that database where we have per plot stored the different treatments of the plants, of the plots. So.
Okay, thank you.
Okay.
So next we have David Bullock, who's hopefully going to give us some more insight on the precision ag side of the same type of data.
Thank you. I'm working with Nicolas Martin on this. His name has been mentioned a couple of times. I'm the PI of a $4 million project. We're just starting the fourth year of four-year project. It's got 30-plus researchers. We're at 6 U.S. universities with collaborators in 3 countries. And I can barely touch on what we're doing. I'm very excited to be here because I just hear people say some things where I think, wow, they know some things, a lot of important things we can't do, and I want to collaborate with you. What I'm going to try to do here is show you why you might want to collaborate with us. All right. A yield response function is that yield depends on managed variables, field characteristics, and weather. That's how God makes corn grow. The problem is nobody knows very much about yield response functions. To learn about that F, we need data on Y, which is yield, managed variables, characteristics, and weather. I think that's a decent categorization of the data we need. There's a lot about excitement about ways folks are generating field characteristics data. There's a Veris machine, which takes electroconductivity. People have been talking about getting field characteristics data at the micrometer level. All right? Terabytes and terabytes of data on types of, from sensors. This is all about sensors. Sensors get what I would call C, field characteristics data. I think what we're missing is data on X. X is farm management data, and I don't think it's varied. Farmers in the same region tend to apply the same amount of fertilizer in each way. So we can get all the imagery and sensoring data in the world, but if we don't vary management input data, I don't think we ain't going nowhere. Okay? And here's why. You can't fit curves through vertical lines. Okay? What we're trying to do is figure out how yields respond to managed inputs in different environments. Okay? If you have different environments, but you don't change the managed inputs, you're trying to fit curves through a straight line. And that doesn't work. Okay? So it is great. We need so badly for people to figure out what the characteristics are and map them. But if we don't also vary the managed variables, we can't figure out the interactions. And the interactions are what is key to management. Okay, so here's an idea. There's a field with a sorghum map, soil types. maybe you have, in one part of the 45A part, maybe this is the production function. In the 68A part, maybe that's the production function. You do a little bit of economics and you find out, given some prices, you need a different amount of nitrogen economically optimal in the gray area than in the white area. If you don't know what those curves look like, you can't solve that economic problem. You got to know how the crop results, how it responds to the managed variable, and how that changes over space with different characteristics. All right. The problem is, if you just fly over Champaign County, you're going to find out that most farmers are doing pretty much the same thing. And so you can't, and they're going to get pretty much the same yield. So you don't get variance in that data unless you do something called what we like to call on-farm precision experimentation. So what's this? Our project is about generating X data and analyzing it to get data-intensive farm management recommendations. Okay, scientists have been trying to generate randomized X data for a very long time. They've been doing it in England since 1843. They've been doing it at more plots since 1876. And that involves a lot of small plots and a lot of graduate students who make too much money. All right, so the thing is, what we're doing is we're trying to create this X data very inexpensively. And how are we doing that? Well, there's a 168-acre field. We did a seed rate and a nitrogen rate randomized experiment on it. How do we do that on 160 acres? No grad students, no running around. There's a farmer, and he's putting in a seed rate trial. And he's doing really fast. I'm not going to play this, but what it does is it bounces up and down. It goes 29, 29.81, 30.01. So in other words, the tractor is trying to put down, the variable rate seed applicator is trying to put down 30,000 seed per acre. Boom, he gets into another place, and it puts down 36,000 seed per acre, or tries to. So in other words, we can computer program a big field trial, and then the farmer just drives. Okay? And then there are yield monitors, and we can get the Y data. Okay? And so we're starting to generate a lot of data. This is an outdated, I'm not even going to say that, because, and there's a bunch of things we're doing in South America. But we're in the United States this year, we're going to run, I don't know, I think 60 or 70 trials, full field trials in 9 different states on 4 different crops. So we're doing a lot. It's a lot of fun. The key is we're working with farmers. They're helping us do the research. And so that's a lot different. I think the key to agronomy is on-farm research. And I think that's the direction we should go to get X data. All right. How's that happen? Farmer sends us a shapefile. We upload it. We run it. We have programs run an experiment. That goes back to the farmer. The farmer runs the thing. The data comes back to a central database. The hardest thing we do is try to get the farmer to give us the data. If you guys can help us, and I know you can, beam that data, okay, from the tractor to us, that's going to save everybody a lot of hassle. All right, what do we want to do? We're wanting to give prescription maps. We want to tell farmers how to farm differently in different parts of the field. This is precision agriculture. Precision agriculture has not taken off because nobody knows how corn grows. Okay? And a lot of what you see from Climate Corporation, et cetera, is bells and whistles. And farmers know that, and that's why this hasn't taken off like it was supposed to. All right. What we can do is we can get a bunch of data and figure out empirically how corn on this 2018 experiment responds to nitrogen and responds to things like topographical wetness index. Okay, we got an R-squared about 0.7, ain't too bad, and so we can figure out how nitrogen should economically optimal change over depending on topographical wetness index. Okay. This is something that my friend Alex and Naira developed. What it says is we don't have to just deal with square farms. We can deal with crazy farms in the Palouse. And we have some very smart people we're getting to work with. How much? 2 minutes. All right. Here's what we want to do. We want to run these experiments all over the world. We want to bring a database to the University of Illinois that has all this data and can also automatize the analysis, et cetera, et cetera. And I would really like you all to help us set up the cyber infrastructure that can do that. Now, that cyber infrastructure would also include a lot of characteristics data. So it's not just what we do, it's what all the sensoring people do as well. It's to put all that data together and the weather together with it. Cyber physical systems, program solicitation, 19553, they want to do stuff with cyber physical systems in agriculture. We can get a couple million, $3 million something dollars like that. I'm going to write a proposal. It's due next month. If you want to write it with me and you know something about computers, let me know. Okay? Why not go for this as a group? I think I've got a lot of this written. If we move fast, we can at least get experiments of writing a thing together. So I think we're ready to do more than just a seed grant. Let's go for some money, and worst thing happens, they reject us, and we do better next year. Yes, thank you.
Quick question for David. So Susanna Rockmallow is a student with Praveen Chandra, and I'm afraid I got the order wrong there.
Okay.
Thank you very much.
All right, there we go.
You should be good to go.
All right, thank you. Good afternoon, I think it is now. My name is Susanna Roque-Malo. I'm a PhD student with Praveen Kumar in civil engineering. I'm going to talk to you a little bit today about the focus of our research group, which is critical zone science, and how it advances the goals of digital agriculture. So to begin with, we define the critical zone as the layer of the Earth's crust extending from bedrock to the tops of canopies. And the goal of critical zone science is to understand not only the processes that happen at each of these scales, but the connection between them. It's a truly transdisciplinary project. It's an NSF-funded project that started in the United States, but now has expanded internationally. But the Critical Zone Observatory, the CZO, that's established here in Illinois is called the Intensively Managed Landscape, IML CZO. We research ecosystem or agroecosystem resilience under the influence of anthropogenic activities such as intense cultivation or tile drainage, which alter soil structure, hydrologic flow paths, nutrient availability, and more. At the IML CZO, we've invested a tremendous effort into extensive high-resolution data collection, and thanks to collaboration with NCSA and CSL, over 5.5 terabytes of high-resolution 25-meter flux tower, in situ sensor, satellite, and UAV-derived data is curated, managed, and made available through platforms like Clouder, which was described just before. There we go. An example of some of this data include LiDAR point clouds, like the one shown in the top left, which covers the Allerton Park. We also have in the top middle imaging spectroscopy rendering of vegetation at Allerton. And on the right, the spinning tree there is a specific tree that we've extracted from that point cloud at Allerton to show the kind of detail that we can extract from hyperspectral data. From airborne LiDAR and hyperspectral data, we can provide detailed information about vegetation structure, formation, and foliage distribution. Now, we can scale this up from just a plant or vegetation scale analysis to a much larger scale, like the one shown in the bottom left, which is all of Allerton. And in that graph, each of those circles represents a single plant or tree, and the color corresponds to the species. Now we can, oh goodness, my screen just changed. Anyway, sorry. Now we can expand this not just from vegetation scale, but to a much larger scale and compute predictions of spatial variability in soil organic matter or legacy historic meander paths of rivers or any kind of structures that are going to change the nutrient availability that farmers or anyone would be interested in. We can also do this for, we can also do this for bioavailable nutrients like copper, phosphorus, iron, and zinc. Now, in order to synthesize and utilize all this data, our group has developed DARA, which is a high-resolution, explicit 3D multi-layer canopy model. DARA models the 1D canopy fluxes, 2D surface runoff and carbon and nitrogen transformations, and 3D subsurface biogeochemical cycling and carbon and nitrogen, hydrologic cycling. All of this is in a coupled CPU-GPU coupled computing environment. The kinds of analysis that we can produce with DARA are exemplified here, where this is an aerial view of a field where the black lines are tile drains and there's a drainage ditch running through the middle. And what we can do is assess soil moisture, nitrate concentration, and age of nitrate with respect to depth and time, and then also overlay that on a cross-section of the field itself, where we can actually see the patterns associated with tile drains and microtopographic variability. Now, that was for a 500 square kilometer plot, but we can do this for a much, much larger area, including this example where one grid size is several square kilometers. And we can still pull out information about soil moisture and tile drainage effects and different effects that we see from variability in microtopography in this area. So in closing, our development of meter scale understanding and prediction of critical zone processes in the framework of an intensively managed landscape in combination with extensive, extremely high-resolution data allows us to make better recommendations for sustainable agricultural management practices and promote an ecosystem services approach to the challenges of digital agriculture. Thank you.
Okay, I think we'll move on. Thank you very much. I have more questions for you later. So I'm going to try and do my slides very, very quickly now. I promise I will try not to keep you from lunch for too long. Luda Meinzer already introduced the ideas of genomics. Corn yields and yields of other crops have accelerated in their gains with modern genetics. For most of that time, we didn't have a lot of data. It was gathering the data that was the biggest issue. Suddenly, with DNA sequencing, we have this colossal exponential growth in the amount of genetic data that we have to cope with. And the growth of computer processing performance hasn't kept pace with that. So you can see this curve is tapering off, whereas the DNA sequencing curve is continuing to accelerate at an exponential rate, and it's actually growing faster. So Moore's law is starting to wear out for computers. We have growth faster than Moore's law. It's about Moore's law squared in DNA. And what do we do about this? Well, we cut corners. Right, so what we do is we have a reference genome. We sequence a reference human, a reference soybean, and we line up the little bits of DNA sequence that come out of the sequencer to that reference genome. Here's an example where we have a reference genome, and in the sequence genome at the top there is an insertion. What happens to those little reads that go into that insertion? We throw them away. They don't line up to the reference genome, and as Donald Rumsfeld was concerned about before the Iraq War, there are unknown unknowns, right? So there are things we don't know because we threw those reads away. We don't know that that gene isn't there, okay? And is this something that there's an alternative for? Well, there is, but it's very computationally intensive. So rather than just line those reads up to the reference genome, what we can do is take substrings of those reads and throw them out into graph space, put them in an extremely complicated multidimensional De Bruijn graph in the memory of a very large computer, and then compare those graphs with one another rather than comparing those sequences. So here we individually assemble these graph-based genomes from individual plants, and we compared the graphs rather than comparing the short reads to the reference genome. Does it work? Well, using an early version of this, we looked at the major soybean cyst nematode resistance locus in soybean. We had SNPs from the type of read alignment and other methods that correlated very well with soybean cyst nematode resistance. We could breed for it, but we didn't know what the gene was. We didn't know what the molecular basis was. We published this together with Andrew Bent's lab in 2012. We found that actually a sequence that is there one time in the reference genome, a 32 kilobase sequence, is present from 3 to 11 times in different soybean cyst nematode resistant varieties. What good does this do? Is we could breed for this before using those SNPs. Why is knowing the molecular basis of this useful? Well, we got the gene, which we think is the most important gene in this region. We did something called a yeast-2 hybrid screen. We found proteins that interact directly with that in a lab experiment, and then we used CRISPR-Cas genome editing to edit out the multiple copies of that interacting protein that are elsewhere in the soybean genome. And you can see from this that that has a very large and significant effect on cyst nematode resistance. So we've been able to identify additional cyst nematode resistance genes from the molecular pathway based on that discovery. It turns out that a lot of more genes seem to follow the same pattern. So we've also cloned the major aphid, actually 2 of the major aphid resistance loci in soybean, and now the major protein content locus. They're all these large structural variant alleles. They're all refractory to the kind of traditional genome sequencing methods that get used. And both of those are currently in press. And we're now working with the Center for Advanced Biofuel and Biotechnology Innovation, CABI, and we're bringing the same kind of genome editing technology into sorghum to enhance oil content in vegetative tissues. All right, and the work I've mentioned here was funded by the United Soybean Board and the North Central Soybean Research Project. So I will skip questions, and I would like to invite Germán Bolero to give some closing remarks for the session.
Thank you. I'm going to be brief, but I just want to point out a few things. I think that we are excited. As an agronomist, I feel really excited about the opportunity to working with the north side of campus. We talk about a lot of south and north side of campus. My dream is to continue bringing together, so I'm going to emphasize what Vikram and Matt mentioned yesterday and today in terms of please send us your comments to the white paper and the website for the Center for Digital Ag. In the great tradition of this university, we want to work from the ground up. And this is about the faculty working on these things, and we think our job is to bring the 2 colleges together. We're making a major effort for that, and we're very proud of that. There are a lot of things that we heard in this past 2 days that are very important and that we never thought of that, and collaboration should happen. Hopefully we can do some sort of speed dating on the website so people get to know what they're doing. And but please knock at our door. We want to create those connections. I also want to thank a lot of people that make this possible. Marisa, Christine, Amanda, Barb, that put a lot of effort in the white paper, obviously Matt and Vikram. And I also like to thank Ranbir, Chandra, Naomi, Abe, that was here yesterday for a great presentation, Chris Harbour, that touched on certain points that I think are very important for what we want to do. I think we talked a lot about the research that is being done, but the creation of the pipeline of students that with job opportunities that this environment creates, and this is a major focus of what we are doing. I think I also want to picked it up from what Jen Bernhardt said this morning in regards of the translation of what we're doing. I think that this has an immediate impact, and that translation has to happen. And that translation in this world means, a technology, a startup, something that benefits society. Society is a big component of what we do, and in the tradition, again, of the land-grant institution like this one, so we want to keep that in mind. And with that, I think that the other aspect that was mentioned in these 2 days a lot was the role of extension. And extension is a big component of this campus and of our college. We're very proud of it, and I think there are a lot of opportunities to partnership with extension for a lot of programs that we talked. Anyway, but I'm going to finish with that. I'm rushing. My job now is to introduce my boss, Dean Kidwell. Dr. Kidwell is the Robert Easter Dean of the College of Agricultural, Consumer, and Environmental Sciences. She's been with us a little over 2 years, but she's a native of Danville and got 2 degrees from the University of Illinois in genetics and in agricultural sciences, and then continue her graduate work at the University of Wisconsin. She's a plant breeder, so a lot of the things that we've been talking, she understands, and she's used, and she's really eager about this collaboration. She's a wheat breeder, so I think that the most fascinating thing for me is that it's someone that a lot of the things that we talk from the point of view from the genetics, the use of technology, and the society, she's done it by breeding for wheat in Washington State and producing wheat that ultimately became crackers. So that's the beauty of a lot of things that we do, right? The quality is connected to that, the final product, and actually the impact of society. So with that, I'm going to introduce Kim, and I promise we finally close with this.
Thank you, sir. Herman, we need a building like this on our end of campus. Note to self. So I'm excited to be here. It's really interesting what happens in life. As a geneticist, some of the best 10 minutes of my life in the last 3 months have been the lightning talks that I heard here, right? I don't have enough time to actually immerse myself in my discipline these days. But what's so exciting to me is to see the merge of transdisciplinary worlds coming together to actually conquer the next frontier in agriculture. There's a joke among plant breeders that when you pass away, what happens is God brings up the screen and watches you walk by the one variety that would have solved world hunger, right? When I think about some of the data analytics that are going on and the precision that's actually being integrated in the system, it gives me great hope that that might not happen, that we actually might find that one. Because even in the day when I was breeding wheat, molecular markers were just starting to come on board. And the precision that added to our ability to make gains from selection was really impressive. Now we're in a frontier where we're looking at landscapes and where plants are positioned in fields can be done to the point where you actually can get a lot more of a feel for the genetic potential and the reality of those plants in that space. Having come from the Palouse, which is a wild place to breed plants because of the topography, it's very hilly there. You lose an inch of rain every 10 miles that you go west in the Palouse. It's a little crazy when you think about environmental variation. What I see is a tremendous amount of possibility. And we're not the only ones that are knocking on this door. But we are one of the premier colleges of engineering in the world, partnered with one of the premier colleges of agricultural, consumer, and environmental sciences in this country, and perhaps the world, depending on what rankings you look at, right? I don't think there's any place around where we have the quality of scientists and the collaborators in this space that we have right in this moment in time. And what's been fun for me, I spent a lot of time in an administrative role dealing with problems. What's been really fun is to watch how excited people are about this project. And I give Matt and Vikram a lot of credit for like grabbing onto this and making it happen. You know, this is one of those grassroots projects for me. When faculty see a door open and there's some administrative support for this, people will lean in, stand up, and make it happen. And what you see is a very different dynamic in the conversation when people come at it from different angles. How can you solve some of the grand challenges, bringing your expertise and technology together instead of competing? I have great hope for this project, and I think, you know, every time Herman comes back from one of these meetings, he talks so fast and is so excited. I just like stand and clap. It's good, right? So I'm glad that faculty are excited about this. I'm really grateful to the provost and his staff for leaning into this investment for growth proposal. I think this is how we actually go from great to extraordinary, by seeding resources into things like this to let them grow. What I know about faculty on this campus, with a little seed opportunity, a lot can happen to elevate. And, you know, frankly, it was heartwarming to see a grad student come up here and just mic drop on a really cool project. You know, well done. She's like embarrassed now, which is awesome. You did a great job. I mean, that warms my heart because, you know, your generation, like, you don't know what you don't know. Like, you know, I was just at Allerton. I drove down from a meeting I had there, and I'm looking. I was just up there. I didn't see all that going on out there. You know, I should have paused a little bit more to think about the dynamic of what's happening out in that space. You know, the fact that we can gather those images and interpret that in a really meaningful way is really quite powerful, you know, and I'm excited about where we're going to go next. I think the industry component in this is really important too, because we are training great minds like yours to go into this world and do great things, and we can't do that without good partnership from industry. It really makes a big difference these days. We are not going to solve the ground challenges of the world in an isolated academic environment. We've got to partner better with our industry folks to really solve these problems. And what we see as people come to the table is a lot of hope and possibility. You know, our research park is a gem that people that have worked here forever don't realize is uncommon. It's not like this on every campus, where you actually have embedded in your structure a research park that draws industry to us to help us do amazing things, that give us an outlet for some of our technology and discovery to go into commercialization for decision-making for farmers and for people in industry. The other thing that's a real hotbed about the research park is students can get hands-on learning opportunities in a way that they bring content into context. That is so important to build confidence and translation skills as part of the academic experience instead of something that happens afterwards, after they get a job. So what I've loved to see is industry get excited about what we're doing and showing up to help us actually, again, go from great to extraordinary. This is not a good to great place. This is a great to extraordinary place. You know, we've gone past the good curve in much of what we do. But by partnering a little bit uniquely, we have the opportunity to be the go-to place for people to actually recruit and hire the next generation of folks that are going to lift this off the ground in ways we never imagined possible when I was in grad school, which wasn't all that long ago in time, but technology-wise, man, I am a dinosaur. And I was a pretty good weep reader back in the day, right? Like sometimes I just envy the technology we have now, and I do wonder too what I missed because I didn't have the precision to actually figure out what the fabulous line was amongst the thousands of lines we evaluated every year. I think as a sign of hope for where we're going here, especially as we're building new academic programs that, again, break down the boundaries across colleges, the computer science, crop science major, I think, is one of the most exciting things that we've done in ACES in a long time. To parlay that into a similar degree with animal science is big as well. If we build it, they will come if we tell them about it. You know, so we're on a marketing bent there. But as we've gone out and talked to our industry folks, my first sign that there's really great hope for this is that we just received a $500,000 gift from Climate Corps for scholarships for people in that program. This is the first gift, not the last gift, right? And it's exciting. So for those of us that came into academia because we want to be the torchbearers of new innovation and new technology and hand that off to the next generation. This is one of my favorite projects, and I'm really glad that you all have the opportunity to bring it to life. For an administrator, there are very few days that are greater than days like these when we actually see hope live and start to crunch and start to elevate. So I thank you all for being curious enough to show up and think about ways to do it differently. And as other people talk about doing this, I hope what they see is our coattails as we rise into the space, because that's absolutely what we intend to do. So thank you for spending, a couple days here. Again, like I only made the last couple talks. Matt, you could have gone on for hours and I would have been happy. That's really the stuff that I love. But I'm grateful to be energized about what I heard at the tail end. And again, if you're going to catch a talk and a grad student's going to mic drop it, it's a good day. So well done. Thank you so much for coming.
Okay, we are done. We're about to go to lunch. I just want to say a couple of words. First, I wasn't sure I could talk about the Climate Corp donation for the CS plus Crop Sciences program, but since Dean Kidwell mentioned it, I think we now have the permission to say it's super exciting because it's the first, it's a real world acknowledgement that the CS plus X programs really matter in the world, and CS plus Crop Sciences, I think, is one of the clearest examples of that. So we're really excited about that. I also want to give a special thanks to Dean Kidwell, because she probably doesn't remember this, but in summer 2017, I had sent you email about this idea of having a Center for Digital Agriculture, and your response was, "This is the most exciting email I've received in my tenure as dean." And that was really encouraging, and I think that it just showed the level of support and enthusiasm that exists on this campus. So I really hope that this center is able to spark super interesting collaborative research across the colleges and really establishes Illinois as a world leader, like I said this morning, in digital agriculture. So with that, I want to ask you all to join us for lunch. We're looking at this as a networking opportunity, as a way to find ways to make connections across the two colleges and people that you might be interested in working with, but also to get some food. So come outside and join us. Thank you.